Hello, welcome to all. Here I would like to explain the process of meiosis. We know that mitosis and meiosis are the two main types of cell division found in living organisms. Among this, meiosis takes place in diploid organism during the gamete formation, that is, during the production of male and female gametes. And this meiosis is a process in which a diploid cell divides twice and form 4 haploid cells. That is the concept of meiosis. And the process of meiosis is broadly classified into 2 steps or 2 stages. Among these, one is called meiosis 1 and second one is called meiosis 2. Now let us move into the process of meiosis 1. In meiosis 1, a diploid cell, that is 12 cell, is divided into 2 cells, and that 2 cells are haploid in nature. So here what happened is the diploid cell is divided into haploid cell, that is, here the chromosome number is reduced into half. That is why meiosis 1 is also called reduction division. Whereas when we take the second phases of meiosis, that is meiosis 2, in meiosis 2, these 2 haploid cells, each haploid cell is divided into 2 cells, 2 daughter cells. That 2 daughter cells are also haploid in number like its parent. So here the chromosome number is not reduced into half. That is why meiosis 2 is not an example for reduction division, but it is an example for equational division, that is mitosis. So we can say that meiosis 2 is equal to mitosis. So that is the concept of meiosis. Now let us try to understand the details of meiosis 1 and the details of meiosis 2. So first of all, let us try to study the details of meiosis 1. So meiosis 1 is again divided into 4 phases. One is called the prophase 1, and second one is called metaphase 1, and third one is called the anaphase 1, and fourth one is called telophase 1. This is the main 4 subphases of meiosis 1, that is prophase 1, metaphase 1, anaphase 1 and telophase 1. If we understand these 4 phases, then we will get a clear idea about meiosis 1. So first of all, this prophase 1 is again divided into 5 phases, 5 subphases. Prophase 1 is again divided into 5 subphases. And that subphases are, first one is called leptotein. Stage, and second one is called zygotein stage, and third one is called paccatein stage. Leptotein, zygotein, paccatein, diplotein, fourth one diplotein, and fifth one is called zygotein. These are the 5 subphases come under prophase 1. So after zygote, metaphase 1 will start, then anaphase 1 will start, then telophase 1 will start. This is the main phases which are come under prophase 1. Okay. Now let us try to understand what are the main features or what are the events which are taking place during leptotein, zygotein, paccatein, diplotein diakinesis. Okay. So, before the starting of meiosis 1, we have to understand interphase of meiosis. We have to study or we have to understand the structure of interphase cell of meiosis. So first of all, look here, this is the structure of interphase cell which are going to enter into the prophase 1. So that interphase cell contain two pair of DNA, that is chromatin reticulum or chromatin thread. It is a long thread of DNA. So here the 2n number is equal to 4. It contains two pair of DNA. And we know that one DNA or one chromatin network, chromatin reticulum coming from father and one from mother. Like that, this one coming from father and this one coming from mother. Sperm cell. And this cell contains a very definite nuclear membrane associated, and along with that nuclear membrane you can see endoplasmic reticulum, Golgi bodies, and also centrosome. So here I draw the structure of animal cell. That is why I draw the structure. I draw centrosome here. So this is the interphase cell, and we know that interphase contains three main phases. That is G1 phase, S phase, and D2 phase. So when this cell is passing through the S phase, its DNA get doubles. That is DNA replication takes place. So due to DNA replication, this DNA and this DNA get doubled. That result, that structure can be clearly visible here. You can see here two DNA is present, one pair of DNA is present. Due to the S phase, replication occurs. Due to replication, it gets converted into 4. Like that, it gets also converted into 4. So at the start point of meiosis 1, that is prophase 1, that is lactoting stage. So here in lactoting stage, you can see replicated DNAs or replicated chromatin fibers. So the main features of lactoting is that during lactoting stage of prophase 1, the DNA or chromatin network or DNA start to condense. Their condensation begins. They start to condense and form a highly elongated chromosome. We cannot able to say it is a 100% chromosome. Here the DNA started to condense into chromosome. So the chromosome can be visible but not clear. In lactotic state, we can see the chromosome, but it is not clear because the condensation is not completed here. That is why we can say during lactotic state, chromosome can be visible but not clearly. Here the condensation just begins. So here you can see long thread of DNA start to coil and form a not well organized chromosome. It's a chromosome but not well organized. So here you can see. That is the main features of lactoting. And when we move into the second phase of prophase 1, that is zygote. Second phase is called zygote. Zygote, condensation of DNA continues. That is why the structure begins slightly changing into a definite chromosome-like structure. So in zygote stage, when we move into the zygote, this is lactoting stage and this is zygote. When we move into the zygote stage, condensation of DNA continues. Condensation of DNA continues. That condensation of DNA gradually continues and finally formed into a structure like this. So during this structure you can see the pairing of chromosome. That pairing of chromosome is called synapsis. So actually in zygote you can see the pairing of two chromosomes. Pairing occurs between the two chromosomes. That pairing is called synapsis or synapsis. That pairing is called synapsis. So synapsis occurs during zygote stage. So this is an intermediate stage between lactoting and zygote. When lactoting is converted during lactoting, DNA undergoes continuous condensation and another phase. That another phase is called zygote. During zygote, you can see this is not a chromosome. This is two chromosomes. We know that this is one chromosome, but we cannot able to see a chromosome like this because one DNA condenses and forms a chromosome like this. But before the condensation, this DNA replicates during during the stage of interphase. That is why you can see a chromosome like this. This is a replicated chromosome. This chromosome contains same copy of 2 DNA. Okay. So, in zygote, pairing between the 2 chromosomes occurs. This chromosome coming from mother and this chromosome coming from father, like this. Okay. That process is called synapsis. That process is called synapsis. And synapsis can be possible with the help of a connecting material or connecting protein which are drawn here by using blue color. That protein is called synaptonemal complex. That protein is called synaptonemal complex. So main important factor or main or important factor here in case of zygote is that pairing between the chromosome occurs. That paired chromosome is called homologous chromosome. This paired chromosome is called homologous chromosome. Like that, this one also gets paired. That paired chromosome is also called homologous chromosome. Here pairing occurs with the help of a proteinaceous connective material that is called synaptonemal complex. Those proteins which help for synapsis, that is called synaptonemal complex. So during all these events, nuclear membrane, Golgi body, centrosome, etc. remains like that. There will be no significant difference in those organelles. And when we move into the third stage, that is called the packaging, then we move into the third phase, that is called the packaging stage. In packaging stage, what happens is exchanging of chromosomal segment between the non-sister chromatin of a homologous chromosome. That process is called crossing over. That process is called crossing over or recombination. This process can occur with the help of an enzyme that is called recombinase. And you know that this is one homologous chromosome, and this homologous chromosome contains 1, 2, 3, 4 chromatids. Among these, 1 and 2 are exact copy. That is why 1 and 2 are sister chromatids. Like that, 3 and 4 also exact copy. So they are also sister chromatids. But 1 and 3 are not exact copy. So they are called non-sister chromatid. Like 1 and 4 are also not an exact copy. So they are also called non-sister chromatid. Okay. So here, in case of packaging, crossing over occurs, which indicates that exchanging of chromosomal segment between 2 and 3, that is non-sister chromatid, exchanging of chromosomal segment between the non-sister chromatid of a homologous chromosome is called crossing over or recombination. That process takes place with the help of an enzyme called recombinase. So during that crossing over, you can see an X-shaped structure called the chiasmata. Okay, but it cannot able to clearly visible in this stage because all those areas are pasted by, all those areas are occupied by synaptonemal complex. That is why the X-shaped structure cannot able to view clearly. When we move into the diploting, another stage, diploting stage. When we move to the diploting stage, here I am not drawn the diploting stage. When we move into the diploting stage, only one event occurs. That is synaptonemal complex. It is a protein that synaptonemal complex slowly disintegrate or dissolve. That leads to the clear visibility of X-shaped structure that is called chiasmata. So during diploting stage, synaptonemal complex dissolves and an X-shaped structure called chiasmata can be clearly visible. That is the stage of diploting. And when we move into the last stage, that is called diakinesis, in diakinesis, chiasmata, this X-shaped structure called the chiasmata completely disappear and the chromosome will be like this. It connects, chromosome will be like this. So they exchange their chromosomal segment completely. So chiasmata terminate or terminalization of chiasmata occurs. So here, like this. Chiasmata terminate. This is the main events which are taking place during the prophase 1 of meiosis. Now we have to study metaphase 1, anaphase 1, telophase 1 and cytokinesis, etc. After that we have to move into the next stage, that is meiosis 2 also, which will be discussed.